Okay, well, welcome everybody, and it's my pleasure to introduce this evening's speaker. Before I do, I just wanted to remind everybody that the lecture will be broadcast live on YouTube, and that's true for the lecture as well as a questions and answers session. So if you're slightly shy or you don't want to be broadcast to the rest of the world, then maybe if you save your questions for after the talk and the questions session and for the drink session that happens at the end, then that might be a good thing. So it's my absolute pleasure to introduce this evening's guest speaker, Professor Geoffrey Armand. And Geoffrey is an Oxford Martin School visiting fellow with the Oxford Martin program on vaccine design and development. And he's also a visiting fellow at my department, the Sir William Dunn School of Pathology, where he's been for the past 2 years. Prior to that, Jeff was the Vice President and Head of Research Discovery at Sanofi Pasteur and headed up all their external research and development programs. And Sanofi Pasteur, as you may well be aware, is one of the few major vaccine companies which is still in existence following acquisitions and mergers of a lot of companies over the last few years. Before Jeff moved to Sanofi Pasteur, he had what can only be described as a stellar scientific career. And he had a 2-year postdoc in Vienna before he was recruited as a lecturer at the University of Genetics, which was a really exciting place to be at the time. We've spoken about the discovery of microsatellite markers in Leicester that time. And he spent just 6 years as a lecturer in Leicester, moving rapidly up through the ranks before he was recruited at the age of 34 to be professor of microbiology at the University of Reading. And the sort of things which Jeff did in his career, which led to that very rapid rise, was that he was the first to show that influenza has overlapping genes. He actually defined the genes in influenza which mediate species specificity. So you may be aware that influenza not only infects humans, but also has very important animal reservoirs such as pigs, birds. And Jeff showed which gene was responsible for allowing viruses to infect specific hosts, and that's very important in terms of genetic reassortment and the emergence of pandemic strains. He also produced the first genetic map of influenza A, and then having done so much on influenza A, moved on to work on poliovirus and started to define some of the epitopes which are recognized by neutralizing antibodies which can actually block infection of cells by poliovirus. So Jeff had this stellar career in academia, was awarded the Fleming Medal by the Society of General Microbiology at the time, and then decided, presumably a change of life, a change of challenges, to move to Sanofi Pasteur back in about 2001, where he was until a couple of years ago, when he came back into the University of Oxford. And since then, his services have been really in great demand. And he sits on the Scientific Advisory Board of the National Institute for Biological Standards and Control, the body which looks after all the vaccines which are released for use in the UK. He's also on the external scientific panel for a program on emerging infections at the Institut Pasteur. He's on the scientific advisory panel of the Purbite Institute, which is interested in animal health. I could go on, but enough is enough. So Jeff has been a man who, despite moving away from Sanofi Pasteur, has not got any less busy, but is in great demand for his advice in the UK. And hopefully, I think one of your goals when you first came back was to try and galvanize vaccinology research and collaborations between academia and industry in vaccine research in the UK. So with that introduction, to give you a background of Jeff, an absolute pleasure to hear what he has to say about are we in control of infectious diseases in 2015? I hope the answer is no, but thanks, Jeff.
Thank you, Chris, for those kind words. The answer is sort of no, but we're in a lot better shape than we used to be in terms of infectious disease control And that allows me to, I guess, to introduce the presentation, because I will mention briefly where we used to be, that is, in those days of history where there were great plagues, and then really what happened as we came into the modern era and we began to get a handle on infectious disease and the agents that cause them, and how much we've achieved really in the 20th and 21st century. And then, of course, there are ongoing challenges, as you're no doubt are aware. So I thought I'd end up by mentioning some of the newer technologies that help us to be optimistic about the future in controlling infectious disease. And there is really an impressive range of new companies and new techniques that are being applied. So here goes. So there are references to infectious disease going way back. Egyptian mummies that show evidence of pox that might be related to smallpox infections. There are carvings of figures with withered limbs, which might refer to poliomyelitis and paralysis. This one is a translation from Hippocrates on hepatitis, and this dates from around 400 BC, of course, and it makes a pretty reasonable description of the symptoms of hepatitis, with its dark-colored bile, which is vomited up, and shivers and fever and pain around the collarbone, etc. So quite likely infectious hepatitis was occupying the physicians of that time. Thereafter, of course, plagues, and particularly the bubonic plague, and the outline of the bubonic plague as seen on this slide is the one from around 1345-46 through to 1353, where as many as an estimated 75 to 200 million people across Europe were killed by this plague. It took 2.5 centuries before the population levels recovered to what they were before that plague. Earlier than that, there was the plague of Justinian in 541, which really devastated the Eastern Roman Byzantine Empire. And then since that time, there have been outbreaks, including 1665 in London, of course, and others in the 1800s in China and India, with quite remarkable and impressive virulence and death rate. At that time, of course, we thought that infectious diseases were spread by bad air, the so-called miasma theory of the origin of these diseases. And this explains the plague doctor's costume. This is Dr. Schnabel von Rom. And in the beak of this uniform that he wore, one might put sweeter smelling fragrances to clean up the bad air that was thought to be behind the transmission of infectious diseases. So in there you'd put mint or spices or camphor or even sponges with vinegar on there. And of course, the plague did give rise to these images, hell on earth, the nightmare here depicted by the Flemish painter Peter Bruegel, which was the triumph of death, reflecting the social upheaval and terror that followed plague that absolutely devastated medieval Europe. It is still with us. These are the figures of plague in the United States during the last 15 years. Small numbers, thankfully, but it is still there. And that's interesting to note. The other scourge of the time was, of course, smallpox, and this slide shows the severe form of smallpox, where with confluent pox, the death rate could be as high as 50 to 60%, and also high in children. And although there were variability in the outbreaks in terms of the case fatality, it really was a devastating disease. I'm sure many of you in this room are aware of the incredible work done by Edward Jenner in combating and eventually leading to vaccination against smallpox. He wasn't the first. There was John Fuster and others, including Jesty and Jensen, around that time. But what Jenner did was he showed that transmitting cowpox to a child, and it is the name of James Phelps, could protect that child when challenged with smallpox. And so the process of vaccination was so developed in 1796. And as you're probably aware as well, the use of vaccines against smallpox resulted in the eventual eradication in 1979, and the World Health Organization was able to declare smallpox zero, or smallpox is dead. One of the fantastic achievements of modern medicine to actually eradicate this disease from the earth. There is another example in the veterinary world with rinderpest, and of course there is a peace dividend that no longer do we have to be vaccinated against smallpox in this situation. So, of course, Jenner didn't really know what he was dealing with in terms of infectious disease and the agent that was causing it. And that branch of microbiology started somewhere else. Actually, 1684, Anthony van Leeuwenhoek described what he called animalcules, small, what we now know to be microbes, which he got from the scrapings around his teeth. And so began the first description of microbes, and then the development of the germ theory of disease came from that, particularly with the work in the mid-1800s by Pasteur, Louis Pasteur and his team. Shown here in the center sitting is Pasteur, the chap fourth from the left, stood up on your left. Sorry, fourth, yes, is someone who made a fortune out of it. But, you know, and I'll come back to him in a moment, because I eventually worked in his company. But for the first time, with Pasteur's inventiveness and the ability to identify, isolate, and observe microbes, we got proper descriptions of bacteria and some of their characteristics through that work at that time. And this just depicts what an old document from that period. What Pasteur also did was he debunked the theory of and that's, I think, one of his major philosophical contributions, debunked the idea of a spontaneous generation of life forms, because at that time there was great debate about whether life could spontaneously generate in puddles and the like, and in the putrefaction of goods, and notably of milk and of wine and other things. And Pasteur showed through quite careful experimentation with his so-called swan-necked flasks that indeed spontaneous generation was not occurring, and that life begat life, and you needed to contaminate a sterile culture fluid in order for it to putrefy and for organisms to grow there. His main contributions, of course, were development also of vaccines, following on from the work of Jenner, and particularly cholera and rabies vaccines, shown here in this slide, vaccination against rage or rabies. The chap who was astute in that panel of figures in Pasteur's, sitting around Pasteur's table, was Marcel Mérieux, who established in 1898 his own laboratory for the productions of serums for the treatment of certain infectious diseases. And I mention him because the legacy of Sanofi Pasteur that Chris mentioned as the company that I worked for as head of research goes back to Marcel Mérieux, one of the Pasteur team in those formative years. And this slide simply points out the history of the development of the companies around Marcel Mérieux from that original founding laboratory. And on the top you have Sanofi Pasteur, which is now the biggest producer of vaccines for human health. And on the bottom there you have Mérieux, which is again one of the biggest producers of veterinary vaccines for animal health. So the contribution of the Pasteur team and those derived from it was huge. And of course, the Mérieux legacy was not just to understand the science, it was actually to make and produce and deliver vaccines that really had an impact on people's lives, and through that enterprise, of course. And of course, the main headquarters and production facilities of both Mérieux and Pasteur, of course, still sit in France, notably in the Lyon area. The understanding of the germ theory of disease, that indeed it wasn't about the bad air that you breathe, but it's about the organisms that infect you. Improvements in hygiene, particularly related to people like Semmelweis and John Snow in London and Joseph Lister, meant that infectious disease became under increasing control. And this slide simply points to the decline in mortality rates due to these 5 diseases: measles, scarlet fever, typhoid, whooping cough, and diphtheria, through to the modern era. And dramatic reductions not only in deaths, but also in the transmission. People became protected, people were aware of ways to avoid infection, and so on. But perhaps the biggest contribution has been vaccines. And today we have vaccines against around 24 different human infectious diseases. They're listed here, and we have one against the eradicated disease, smallpox, at the bottom here. And it's without doubt that vaccines have been of enormous value in bringing infectious diseases under control. Probably the greatest public health triumph after improved access to clean drinking water. More than 3 million lives saved every year through the use of vaccines, but we still don't quite do enough, and there are still people dying of vaccine-preventable diseases around the world. But the value is there. Really, not only smallpox being eradicated, polio infections, and I'll mention the eradication of polio a little later, but fell globally by 99% since the campaign to eradicate polio was started in 1988. An estimated 5 million people have escaped lifelong paralysis as a result of that campaign. Measles mortality dropped by around 75% between 2000 and 2007, with increased campaigns to use measles vaccine. And the story goes on. Here are some of the impressive curves. This is the reduction of polio incidence in the United States, 1920 to 1972. Here is the impact of transmission of Haemophilus influenza type B vaccines in Canada in the more recent period, 89 to 2004, where uptake of vaccine meant that transmission was blocked, and the risks to uninfected people are, of course, dramatically reduced as a result of that. And this slide simply translates the pre-vaccine era approximate morbidity and the most recent reports in relation to those diseases. So you see diphtheria is more or less dealt with 100%, Haemophilus influenza 99%, hepatitis A 91%, and so on down the page, as you can see. So dramatic reductions in the incidence of infectious diseases as a result of vaccination. This led Neil Ferguson in his book, Civilization, which I enjoyed reading a year or two back, to really identify and highlight vaccination as one of the major medical advances that has contributed to human civilization. It's meant that we're more confident in where we travel, we're more confident in planning for the future, we're more confident in bringing up our children, and we have all, as a society worldwide, benefited enormously from its widespread use. And these are just some figures from, or some observations from Gavi, the Global Alliance for Vaccines and Immunization, and which has the objective of protecting health and improving education and boosting prospects through vaccination. It is a virtuous cycle, as pointed out by Gavi and its advisors. Children vaccinated from birth to 2 years are likely to be healthier and live longer. They have fewer serious illnesses, lower healthcare costs, more family money available to spend on improving their life. the child attends school better, there's a better economic outlook, the birth rate may drop. As a result of that, community is more economically stable and productive, and there's more politically unstable economic countries as a result of improvement in healthcare that immunization provides. So it's really a remarkable virtuous cycle that has helped a lot in bringing countries out of poverty. It's clear that protecting children from infectious diseases raises IQ. It improves cognitive function. It keeps them healthy, improving their time to learn, improves their productivity, and it frees the family from crippling medical costs. It also, with healthy children, leads to a declining birth cohort, which means that families are generally more stable and healthier. An estimated 30 to 50% of Asia's economic growth from 1965 to 1990 is attributed to reductions in infant mortality and fertility rates. So spending on child health is one of the greatest impacts on improving lifetime earnings for members of the society. Gavi's work in this is remarkable, and it continues to promulgate vaccination and organize the purchase and distribution of vaccines from the big companies, often at real knockdown prices. And working in the industry, it was always a bit of a battle with Gavi because they wanted to grind you down to zero profit on the products. But nevertheless, their impact has been remarkable. This year, they expect to be their peak year for the supported introductions of vaccines in different countries around the world. Listed here, I don't have time to go through them, but the various vaccines that they've pushed into various parts of the world. Number of deaths averted just since 2010 is getting on for 4 million and rising. If you're British, I think you can feel reasonably proud of this particular figure, which points out that the United Kingdom is by some substantial margin the biggest contributor to Gavi activities in the provision of money to buy vaccines and the pledges as we go forward. Quite significant, even compared with the United States, with France, with Italy and Australia. You can read them, and up the second column they get even smaller. So at $2.4 billion, this is a substantial contribution, and it's, I guess, where the 0.9% that David Cameron refers to in terms of his overseas aid, it's where at least some of it goes. So I think something to be quite proud of. In spite of those advances, and in spite of Gavi's excellent work in taking vaccines into developing countries, there are still challenges. Dengue fever threatens at least 2.5 billion people. We don't quite yet have a vaccine, although I will mention something later on. We're almost there. And then some of the people infected by that dengue, I should say, those dengue viruses, as there are 4 of them, go on to develop severe hemorrhagic fever, which is a particularly nasty form of the disease. Don't forget seasonal influenza through, let's say, a less than ideal vaccine, also strikes about 600 million people. In fact, there's a recent calculation based on US figures suggesting as many as 1 billion people a year develop influenza. And some of that is very severe, leading to somewhere in the region of 250,000 to 500,000 early deaths through influenza and associated then secondary infections. And hospital-acquired infections, particularly of antibiotic-resistant organisms in hospital settings, is nowadays a major concern for public health in many industrialized countries. This little chart from just a few years ago in Nature makes the point that worldwide, respiratory infections in particular, but infectious diseases in general, you see the list there on the right-hand side, contribute quite substantially to worldwide deaths. So we are not yet in control. The threat of these diseases is not yet over. And coming back to the Gavi objectives, This was the figure for 2014, an estimated 18.7 million infants are not fully immunized with the diphtheria, tetanus, and pertussis vaccine that is really the mainstay of childhood vaccination. And you see the regions of the world which are most lacking in this regard, particularly Africa and Southeast Asia. So, what are the ongoing challenges then in our control of infectious diseases? Access to vaccines, yes, and Gavi is doing a lot, as I've just mentioned. Middle-income countries, the ones that fall outside the Gavi domain, tend to become also at risk because once they have reached a certain GDP per capita, they're no longer eligible for the Gavi supply through the philanthropic route that Gavi provides. And those middle-income countries become responsible for their own healthcare budgets. Vaccination tends to fall. It's interesting, and it's important that those countries are able to find the resources to maintain the vaccinations that Gavi has often initiated and got moving on. There are also things like anti-vaccine groups that are a barrier to the uptake of vaccines. A little more on those in a moment. There are other challenges like how can we build on smallpox eradication and work out further eradications, and what are the challenges there. There is developing new vaccines against what we call the difficult pathogens, those pathogens where they play tricks on us, they evade our immune responses, they show incredible genetic diversity, or they somehow are able to hide away from our immune systems. These include things like HIV, HCV, hepatitis C virus that is, tuberculosis, malaria, and opportunistic bacterial infections and so on. Then there are the ones that strike just occasionally, where it's difficult to get all the resources together to have a real push at developing a vaccine because these things come and go, and we're never quite sure how much time to spend developing vaccines against a potential future attack. Into this group go Ebola, MERS, SARS, chikungunya, these types of infections that are often of the developing world, and again, as I've said, rather difficult to galvanize the developed world into taking seriously in terms of vaccine development. There are emerging pathogens, and some of those I've just mentioned are also emerging pathogens. There's the problem of antibiotic resistance. There's the problem of bioterrorism and the potential deliberate release of some of these organisms. So just to touch on some of these points as we go through the next 20 minutes or so. MMR, a jab too far. This was an unfortunate piece of publicity given to an unfounded assertion that MMR was causing autism. The evidence supporting that was never there. It's very difficult to prove a negative, so it's very difficult to get confirmatory evidence of absolutely zero risk, but that's not the point. There was no evidence in the first place to somehow associate MMR with autism. Perhaps the most difficult point for the mothers who suffer with children who develop autism was the timing. Autism is generally diagnosed in the sort of 18 months to 2-year time frame of age. MMR happens to be given at 15 months. And so temporally there was that association. The reason MMR is given at 15 months is because it's a live vaccine. Those are weakened strains, attenuated strains that infect the child. If you give them earlier, there are enough maternal antibodies still in the child that the vaccine doesn't properly take and doesn't immunize. So you've got to delay it. So it goes into the second year, and that is the timing where, as I've said, autism may be recognized. But the association between the two and the assertion that MMR causes autism was unfounded. Nevertheless, the publicity that it caused saw a reluctance to vaccinate, particularly with MMR, and a resurgence of measles in the sort of late 1990s and early noughties that is even now difficult to control. Even in the United States, this same sort of publicity has led to a fairly dramatic increase just last year in 2014 in measles cases, and again, real concern that as soon as you hesitate for a moment in vaccinating against some of these highly infectious pathogens that spread around very, very easily, then these diseases can return. Okay, so I've commented on the impact of vaccination, but the control of infectious diseases, of course, is not just due to vaccination. We can't mention infectious disease control without mentioning this chap who worked in Praed Street in the 1920s and 30s, and of course first discovered penicillin, and then it was Florey and Chain and Abrahams who worked on that original discovery to develop ways of producing penicillin and using it in the clinic in a meaningful way. And of course, that too heralded an incredible period of control of already infected people and treatments which saved millions of lives. And of course, there was that heyday of the discovery of antibiotics following Fleming, such that there were many new antibiotics discovered in the 1950s and 60s and brought to market all through the 70s and into the 80s. But there's been a relative void of discovery of new antibiotics since that time. And of course, we've begun to see, in a big way, resistance to antibiotics. Major classes of antibiotics are listed on this slide with their approximate timings of discovery. But there was such a period in the 1950s and 60s that because all of these antibiotics were available to us, because the vaccines were having such a dramatic impact on the incidence of disease, that the United States Surgeon General, Dr. William H. Stewart, almost made this statement, which came back to haunt him, which is, it's time to close the book on infectious diseases and declare the war against pestilence won. It was premature, and we now see problems. First of all, our ability, it seems, to develop new antibiotics is dwindling. These are the numbers of new antibacterial agents in the various time frames listed on this slide, and you see it's getting down to not very many by the time we take the last 5 or 6 years into account. There are economic reasons for that as well as scientific ones, and it's interesting that the cost of drug development is such that the incentives to develop new antibiotics are really not there from a commercial point of view, and we can talk about that later if you wish. One of the things, though, is that there has been the older antibiotics tend to be of less value as time goes on. We've sort of overused them a bit, not only in treating human infectious disease, but also in doing things like putting them into animal feeds to ensure more rapid and disease-free growth of animal stock for feed. And so antibiotic resistance has become a big problem. Resistance, you'll find it everywhere. A lot of these products, the antibiotics, are natural products that are made in wars between bacteria that live in the soil, or wars between bacteria and eukaryotic protists that live in the soil. And wherever you look, you'll find resistance mechanisms to antibiotics that exist out there. The key is, though, how do they get spread around, and what happens? Why do bacteria that were once sensitive to particular antibiotics become resistant? You see the speed here of the detection of resistance, and then it becomes widespread and gets spread around. Better insights into what's happening and how the bacteria evolve have started to happen. This is a study by the Institut Pasteur, François-Xavier Weil, and others from the Wellcome Trust, as part of a major consortium looking at Shigella dysenteriae, which causes a severe infection of diarrhea, large outbreaks, particularly problems in war zones and conflicts, and famine, and wherever you have refugees, these organisms can spread around. And Gallipoli was one of the first major appearances of this disease in the First World War, where many troops were struck down by Shigella dysenteriae. And what we can do now with DNA sequencing, rapid DNA sequencing, is look at the organisms that have been isolated from various parts of the world over the last 100 years or so, and really begin to map the evolution of these organisms. Where have they come from? Where have they spread to? How have they acquired antibiotic resistances, and so on? And the studies that this group has done has identified European lineages, global lineages, South Asian lineages of bacteria, and has been able to understand the relationships between them. But perhaps the most important practical is the accumulation, as you see in red here, of antibiotic resistances in the various groups of bacteria that have been isolated from different parts of the world. And perhaps this bottom graph here is the most telling, which is the accumulation of antibiotic resistance genes and genetic mechanisms to allow them to overcome streptomycin, sulfonamides, tetracycline, chloramphenicol, ampicillin, etc. A dramatic ability of these organisms to capture DNA that encodes resistance mechanisms and pass these DNAs amongst themselves, so that the traits with antibiotic resistances become to predominate as antibiotics are used. So antibiotic resistance has become a particular problem, and one we can perhaps return to right at the end when we talk about new technologies. Coming back to control of diseases in general, I was quite thrilled to see this in The Economist just a couple of weeks ago, in fact, pointing out that eradicating disease does save a lot of lives, and that there are a number of diseases which are eradicable with, shall we say, just a bit more of a push from World Health Organization and health authorities around the disease around the world. But The Economist pointed out that eradicating diseases is not only a good thing, it's also a good time to do it, because a lot of diseases are at the point where a push will get rid of them. The list is on this slide, and I don't expect you to read them all, but just to say things like the guinea worm disease and yaws are somewhat obscure, but you see in there this polio, measles, mumps, and rubella as potentially eradicable from the earth if we vaccinate and prevent transmission adequately. And this is rather remarkable that we can get there. And I just like to give you a little journey into polio, which Chris mentioned I worked on in my academic career. And I was involved in understanding how polio vaccines work when the World Health Assembly in 1988 announced its push to try and eradicate polio from the earth by really pushing vaccination, and particularly its main tool has been the oral polio vaccine, the one you take on a lump of sugar or a drop into the mouth, which is actually a mixture of 3 live strains which grow in your gut but have lost the ability to infect the neuron and cause paralysis. The use of those vaccines, and there is an alternative one which I'll come on to as well, the killed vaccine, which is the injectable form, those have been enormously important in reducing the incidence of polio in the way that you'll see. First to say that you see the map of the world in red of where polio still was endemic in 1988 and in 2014, and this year we have just a little bit in Afghanistan and Pakistan and Nigeria. The polio eradication, the numbers are here. In 1988, when the World Health Assembly passed its resolution, we were still in the 300 to 400,000 cases of paralysis per year. That has dramatically declined, and we're now in a situation where there are a handful of cases, but they're becoming increasingly difficult to mop up. And a little story about why this may be the case. There is a guy who has been followed now for 28, well, he's been followed for most of the 28 years, but it's estimated to be 28 years since this chap was vaccinated with the oral polio vaccine. I mentioned it's alive attenuated, it grows in his gut, and this gentleman has an immunodeficiency, so he doesn't make antibodies. His health is dependent on him having monthly injections of immunoglobulin from donors, but he lives a pretty normal life. He lives not so far from here, actually. I'm not allowed to identify him, but it's interesting the way he has continued to shed polio. Generally, it also should be pointed out that what are called circulating vaccine-derived polioviruses, that is, they come from the vaccine itself having mutated and reacquired virulence, are a bit of a concern. And this map gives some small outbreaks that there have been, and maps them geographically for both polio type 1, type 2, and type 3. There are 3 different types of virus over recent years. And chronic poliovirus excretion, as this chap I was referring to, is happening. There have been at least a couple in the United States, one in Germany, a couple in the UK, and often these people shed virus for a number of years, and the one I referred to is still shedding after 28 years. And these people have all immunodeficiency, usually the common variable immunodeficiency, which means their antibody production is lousy. And the interesting story is this particular gentleman, the 28-year man, I shall refer to him as, colleagues at the National Institute of Biological Standards and Control have tried to ask, can we cure him? Can we get rid of his virus? And drug use hasn't worked. The virus in this chap's gut is not completely sensitive to any of the known anti-picornaviral agents. So they tried giving him immunoglobulin. That didn't work either. And they said, what about maternal milk, human breast milk, in other words? This chap is in his 30s, so he has, for these periods, human breast milk on his cornflakes. And it sort of works a bit. And you see there in about the 2001 time frame that his viral load, this is the amount of virus in his feces, which can be measured on a daily basis, actually. And you can see this is logs of virus, so this is 1 million viruses per gram of feces of this chap. During the first maternal milk feeding, it dropped down. Actually, there's a bit more detail on the next slide of the individual feeding. And the blue batch of mother's milk did rather better than the red batch of mother's milk in bringing the titer down in his feces. Unfortunately, however, stop the mother's milk and the virus bounces back, so it doesn't actually cure him. And fortunately also, this chap is currently shedding, and he is shedding, but he's shedding into a fully vaccinated community, so everyone else is protected, and there's no real danger for contacts of this person. That said, of course, if we move into a polio eradicated world, we can't stop vaccinating if there are people like this guy around. And it's interesting that the more we use the oral polio vaccine, the more likely we are to create these sort of people with ongoing persistent infections of polio in their gut. Why is this of concern? Well, it's vaccine virus in his gut, yes, but it changes, it evolves, and we can even map a rate of change as the virus is growing in the gut of these individuals. And there is something like a 2.8% change in the base sequence of the genome of the virus per year. what this means is that, and you see this particular chap is, I think it's the blue, yes, this one, the blue. So I can't see the colors very well. I think it's this group and he's now diverged somewhat away from the vaccine in terms of the nucleotide sequence. This is important because you accumulate changes on the surface of the virus, which means the virus that is shed by this guy are not quite as neutralizable as regular wild-type viruses, so it potentially could escape immunity. And the worst observation is that actually it's a lot more virulent. The natural Sabin2, which this strain that he's shedding is derived from, you need about 10 logs of virus, that's 10 billion viruses, to paralyze a mouse. With these excreted viruses, you need 10,000 times less. So it's significantly more virulent, the virus coming out in the feces of this chap, than the vaccine that went into him in the first place. All right, so long periods of poliovirus excretion from this individual really then is a problem. Vaccination we cannot stop after so-called eradication while these long-term excretors are still there, and we will need to vaccinate long after global eradication. So for this virus, for this infectious disease, no peace dividend, we can't stop vaccinating. There will be countries who will say, well, why should we keep vaccinating when the disease is gone, particularly those middle-income countries that are strapped for cash and healthcare costs. But it will be very important to maintain a polio-free world. Quickly, just to come through a few more things. There are new vaccines, of course, we'd like to develop many more. Approximately 25 have been developed, as you saw from my chart earlier. There are about 70, so 40 or 50 remain for which we'd like to develop new vaccines. But they do show antigenic diversity. There may be limited natural immunity. They may have a role in where the immune response contributes to the pathology. In other words, they're quite difficult. We don't know what immune responses to stimulate to develop vaccines against some of these more difficult pathogens. The easy ones have been done. The remaining ones are difficult. Here is a list of those that you might like to develop. I don't have time to go through it, but it's quite an intimidating list. Only some of these, of course, are globally important, worldwide, and high incidence numbers, but they represent a considerable biological challenge. The other thing is the cost and the time. To develop a new vaccine today is estimated to cost about $1 billion. And takes in the region of 12 to 20 years, depending on quite where we start and how long the clinical development takes. But I don't have time to take you through all of this, but just to say that a lot of work is done outside the pharmaceuticals industry by academia in understanding the infections, but the work of developing the product and its testing, safety testing, and then clinical testing, and then of course scale up for manufacture and supply and registration and launch is done inside the industry. And it's this part that takes in the region of $1 billion. Some specific challenges among that list are things like respiratory syncytial virus, biggest cause of hospitalization of children under the age of 6 months. Seasonal and pandemic influenza, the vaccines are not as good as you would like, and in a pandemic scenario, we're lacking. Rhino and other influenza-like illnesses, cytomegalovirus, the biggest cause of fetal abnormality, herpes simplex virus, recurrent sores, cold sores, and so on, as well as neurological invasion, Epstein-Barr virus, and then the antibiotic-resistant bacteria. There are a wide range of them associated with diarrheal diseases, methicillin-resistant Staphylococcus aureus and other nosocomial infections. And then we have the multi-drug resistance and the extensive drug-resistant tuberculosis strains, which are a real problem. After that, you have chronic infectious diseases where people are already infected and you like to be able to vaccinate to cure the infection. That applies to things like hepatitis C virus, hepatitis B as well, big problem in China, HIV, of course, cytomegalo, again, HSV and TB, and then emerging pathogens. Emerging pathogens, it's always with us. Things are popping up all the time, facilitated by things like commercial air travel, global trade, urbanization, unchecked population growth, and climate change. Again, I don't have time to go through this map of the world, but just invite you to see the wide range of infections that have emerged in the modern era, including Ebola, which I'll come to in a moment, but also things like Nipah, Hendra enterovirus 71, hantavirus, etc. And the substrate, that is the breeding ground for these infectious agents, is us. And I needn't remind you of the incredible growth in the human population in the modern era, to the point where we're now almost 3 times as many people in the world as on the day that I was born, and heading up swiftly to 9 billion. And that's a great substrate if you're an infectious agent that lives in humans. There's plenty of us to go at and to get your teeth into. Some of the scary things are things like Ebola. You're all aware of the Ebola outbreak in Western Africa starting in 2014. It dramatically escalated in numbers. This is through 2014. It particularly infected Sierra Leone and Liberia. The current status, and this was just the WHO website a few days ago, said that there were no cases week beginning 14th of October and 7th of October, but 21st of October, 3 new confirmed cases, so we're not completely on top of it yet. The total tally is somewhere in the region of 28,500 cases with over 11,000 deaths. The worldwide response to this has been impressive. I was impressed by the Gavi example earlier of companies pledging money and the incredible work that Gavi has done, but so too against Ebola. Over $500 million with World Bank, United States, Britain not sluggish in this either, doing quite well, even more than the European Commission, which is remarkable. And really quite good intervention here has been developed very, very quickly. The best results so far have been developed, I think, by this new vaccine candidate called VSV-ZBOV. It's an engineered virus of a cattle virus called vesicular stomatitis, which has been engineered to take the Ebola glycoprotein, and these lines here simply refer to the modified genome now with an Ebola piece in there. You grow the virus, and it now has a bit of Ebola in it. It doesn't cause disease in humans. It has a single cycle of replication in human, but it stimulates an immune response against the Ebola protein. And remarkably, and I just invite you to look at these vaccine efficacy studies, 100%, 100%, 75%, 76%, depending on the group sizes and the scoring that was done. I don't have time to tell you about the particular design of the trials, but these are remarkably encouraging results for Ebola vaccine that have happened in the last 6 to 12 months. To date, there are 15 vaccines that are being developed. The one that I mentioned, the VSV EBOV, as well as another one called the chimpadenovirus EBOV, are the most advanced. Pharmaceutical companies are committed to ramp up as soon as we have good evidence, which has happened, of course, now with the EBOV on VSV. And this could be really the fastest vaccine development and rollout in history. I mentioned earlier the cost of $1 billion, and 12 to 20 years, this has been done in double quick time, and it's really quite impressive. It hasn't been through quite the range of testing that you would normally demand for a regular vaccine, but in the urgency of the Ebola outbreak, it certainly has been a remarkable achievement. So just to finish off that further specific challenges, how do you go about developing interventions against that list that I read out that's here again on this slide? And I don't know the answer to that, except I'm encouraged always by the march of technology. And the new tools and technologies that are emerging offer optimism, I think, that we will be able to deal with infectious diseases for most of what nature throws at us. And of course, it's always cleverer than we think. So we can never be absolutely confident. But just a few last slides very quickly to show you some of the things that I've observed from my role in Sanofi, looking at small biotechs emerging in the vaccine area, infectious disease area. What are the types of things that they're developing, and what can they be applied to, and how good are they? First, vectors, the construction of chimeric viruses. The one in yellow here is the same sort of thing as I've just told you about. This was the Ebola example on an earlier slide. Same thing was done with dengue, so that we now have a chimeric virus, which is really the yellow fever vaccine in its interior, but is now decorated with the dengue coat proteins, 4 different versions of this for the dengue virus. This has been a remarkable development as well. It's taken a lot longer than Ebola and a lot more money, but very likely we will be using dengue vaccine next year, which is based on this chimeric virus vector technology, which looks very promising for some interventions. That's basically what it looks like for the yellow fever and the dengue, where you take the middle of the yellow fever, but the coat from the dengue. Other things in development, not really a vaccine as such, but an aid to development. This is immunology in a test tube, developed by a company called VaxDesign in Florida. They can leukapheresis, take white cells, white blood cells out of individuals, establish them in culture, and actually vaccinate the culture so that you develop both cellular responses and antibody responses in the test tube. And they can monitor those responses as the cells are stimulated with vaccine candidates, enabling you to do some of the rather expensive and lengthy and cumbersome development of vaccines that you had to do in patients, now in a test tube to get a proof of concept and rapid development. You can even do different test tubes with different people. So you can have almost a field trial, a clinical trial, with a couple of hundred different subjects, each with their own test tube, where you're stimulating and you look at the proportion of those that respond, the types of immune responses that they make, and you can work out how best to design your vaccine when you generally go in the clinic. Remarkable development. Here's another one, which is in vitro protein synthesis, allowing you to make proteins under well-defined conditions, proteins that are antigenic, that are immunogenic. And this company called Sutro, and thrown off a vaccine company called Sutrovax, is harnessing this technology for new types of ways of making pneumococcal vaccines, influenza vaccines, respiratory syncytial vaccine candidates, and so on. Yet another one is RNA vaccines, where instead of trying to make the antigen and administer it as a vaccine or develop a live one to feed you or to inject you with, these guys are using RNA directly, and they've worked out ways of stabilizing RNA to get it into your cells so that your cell makes the foreign protein, your immune response sees it, and you mount an immune response. Amazing amount of investment has gone into this company called Moderna, about $1 billion in the last year or two, aimed at developing RNA as a way of delivering therapeutic proteins, including vaccines. How do we deal with antigenic diversity like that in HIV? This is a diagram showing the extent of antigenic diversity of HIV in the Congo in 1 year. Compare it with influenza in 1 year. The breadth of the arms of this dendrogram relate to the amount of diversity. This is HIV in a cohort in Amsterdam in 1991. This is HIV in a single individual varying over 6 years. Dealing with that is incredibly difficult as a vaccinologist. But now structure-based studies, and particularly fueled by knowledge of monoclonal antibodies, which are broadly neutralizing, where do they bind to on the virus? Can you work out, therefore, which are the important parts of the protein to present? And so structure-based engineering of antigens to create something quite new in terms of its overall structure, but important in maintaining some of the structures in the original molecule, as you see here, and using these cut down versions to provoke immune responses that wouldn't normally be provoked by these proteins, which are highly variable on their surface, but have these inner cores of conservation that might provide the basis of attacking the part in all that diversity that is common to the different members of the HIV or the HCV or the influenza population. Virus-like particles. I don't have time to tell you much about it, but these are effectively viruses which are devoid of nucleic acid, being used in various ways, but again, the technology for developing these for epi-simplex, cytomegalo, HBV, papillomavirus are really remarkable, and our ability to make and produce and then industrialize and manufacture in the millions of doses of these sorts of things has grown enormously. I'm sorry, I was on the wrong slide when I was talking, but there you have examples of the virus-like particles. If you can't make vaccines, you can even use these days cocktails of monoclonal antibodies to give short-term protection. Here's a company based in Austria, which, because Staphylococcus aureus is such a difficult pathogen to develop a vaccine against it, are designing a cocktail of monoclonals to protect you short-term. If you go into hospital, you're at risk of meeting one of these highly antibiotic-resistant organisms. Can we provide you with short-lived immunity before you go in? And this is an observation that they've developed antibodies that seem to be able to kill Staphylococcus or E. coli, which are also highly resistant. Another interesting and different intervention is this one here from a small company called Virion Health, growing out of Warwick University, who've worked out that the defective viruses can be grown, and if you administer these defective viruses and they meet the real virus in the individual, they hijack the replication machinery of the real virus and interfere with its development and dramatically attenuate the infection as a result of doing that. This one that they've developed is based on influenza A. It also stimulates interferon, so it seems to be active not only against influenza, but other influenza-like illnesses, the common cold. Here, just to end, is the last one I'll mention, which is a new way of making antibiotics. We now realize through our ability to do deep genome sequencing that we've only ever been able to culture a few % of the bacteria that are out there living in soil, on our skin, in our gut, and in animals, on animals. And more recently, we've been able to see them through DNA sequencing, but we can't culture them. Since most antibiotics are derived from other organisms, it effectively says that we've only been looking in the antibiotics we've developed in 2 or 3% of what's out there. And we'd like to look at the other 98%. This is a company called Warp Drive that are doing in silico searching for what they call gene clusters that make secondary metabolites in bacteria that we're unable to culture. They then pick out those DNAs and put them into an organism we can culture, see what metabolites are made, and then test those metabolites for their ability to attack other microorganisms. And so they claim, and it's early days yet, we have a whole new approach to the development of new types of antibiotics. So that's the last technology that I mentioned, but the last few slides give me grounds for optimism that, we are not in total control of infectious disease, but the tools that we've developed, the incredible progress we've made over the last 150 years, and the incredible pace of the development of new technologies suggests that we can be reasonably confident that we'll deal with what nature throws at us and at least find some response in terms of antibiotics, vaccines, or other ways, antibodies, of dealing with infectious disease. We can never be complacent, as we've seen with the return of measles, if we just hesitate vaccination for just a moment. But I think we're well equipped, and the march of biotech and the march of modern biological methods reassures us that we're in a good place. And I thank you, Chris, for allowing me to run over time a little.
So, Jeff, thank you for an outstanding presentation. Took in the breadth of history, glimpsing into the future, new technologies, and how we benefit from old technologies in the past. Wonderful talk. We're now open for questions. There's a roving mic for anybody who wants to ask a question. Can I start off then, Jeff? I mean, you spoke about this remarkably rapid development of the Ebola vaccine and how that's ready to roll out. But now it's ready to roll out. You also showed the cases have actually dropped off through public health interventions, and it's been extremely successful. How do you envisage any vaccine being introduced into those countries which are not natural reservoirs for the organism?
Well, in the future, at least we'll be ready very, very quickly, and the idea will be to vaccinate around initial cases. You're quite right that our ability just to implement regular public health measures of disinfection, isolation, and generally avoiding contact with infected individuals, it works enormously well when we're well organized. We weren't well organized in the early part of the Ebola outbreak, and it, as you saw, the figures took off. But as you get control, the figures come down. I remind you that when the SARS outbreak happened in 2001, we did get control of that through quarantine only. We had no interventions at all. We had no vaccine, no drugs, but we managed to get hold of that pretty quickly. It's fortunate that SARS wasn't an enormously infectious virus. It required reasonably close contact. So, and some, as you know, some viruses require intimate contact. HIV, for example. Epstein-Barr we used to call kissing disease because it requires exchange of saliva. But things like measles, and particularly foot and mouth in cattle, walk into the same room and you're infected. They're incredibly infectious. SARS was not so infectious. Ebola is similarly not enormously infectious. But we'll be in a better place next time Ebola strikes as a result of the vaccine development that's been going on this last year or so.
Thank you.
Hello. Do you think a universal vaccine based on for flu would actually ever work based on the Stewart region?
It depends on your point of view. I think it's a challenge to get a vaccine that. The problem with flu is that sometimes we have a mismatch because the strain is chosen to go in the following winter's vaccine, usually around February. It's a World Health Organization decision, and that strain is then supplied via influenza reference laboratories to the manufacturers, who start making new vaccine like hell through the summer to distribute in the sort of September, October timeframe for vaccination for that winter. Now, sometimes in that space of time, the virus changes and you end up with a not very good strain in the vaccine, and so it's not very good. But some years it's fine. You get a good match and it's a good vaccine. Now, to have a universal vaccine that is going to last you, 10 years, or it's going to be effective against all strains, not just in the past, but new ones to come. And for it to be as good as a good matched year, I think is a hell of a challenge. It might be better than a bad matched year, but you want to do better than that. Your standard of care that you've got to do better than, I believe, is a well-matched year. And if you're going to do better with a little piece of the antigen rather than the whole antigen, I think it's quite a challenge. But there's a good deal of optimism out there, and we'll see what happens in the next 5 to 10 years in that space.
Hi, thank you. When you were talking about the 28-year man who's still shedding, you mentioned the problem that the virus in him is evolving at 2.8% a year, and that it might escape from the strains that we're immunizing with.
Yeah.
I wonder why you didn't mention similar evolution in the wild-type strain that's still floating around?
Interesting point. Wild-type strains in general, in normal evolutionary circumstances, don't stay in people for 28 years because those people with immunodeficiency die. In the modern era, we keep them alive through his monthly injections with immunoglobulin. So in a way, we've created a new incubator that didn't exist in nature before we were able to treat these sorts of people. And the consequence is then that he gets this infection. We can't quite get rid of it. I should have referred to another slide. I quickly went past it. But we know about this chap. And as I say, we've followed him for quite some time. I think it's now 170 different isolates from him, and you can see how they change. But there's evidence for the existence of these people in Finland, in Israel, and other places. And you find evidence just by dipping the sewage and looking for virus. And what you find is virus that's a bit like what's coming out of this guy. Now, we don't know the members of the population that are shedding it, but it's clearly there. And it's not wild type. It is vaccine derived. So he's not alone. He's not unique. And yeah, these viruses are evolving, and we've somehow got to be able to deal with it. I think probably what you would need is a drug that absolutely stopped them, and then to identify the people and treat them with a drug and stop them shedding. Some do spontaneously stop, as my slide showed. So this one, 28 years, is rather rare, fortunately.
Has he been re-immunised with the oral polio vaccine?
Well, he
really
can't make an immune response, so there's no point.
But in a sense, it might be a competition at the mucosal surface.
Yeah, well, indeed. There is some evidence from some earlier shedders that the thing that stopped them shedding was a good dose of the runs. They got a Shigella and it wiped everything out and reamed him out, and that got rid of the poliovirus as well.
I don't think you get many takers.
But no, I think it'd be a bit harsh to treat him like that. But anyway, it's really fascinating biology and interesting to observe. But as I mentioned, the peace dividend is not therefore going to be there. We're going to have to continue. And you know, countries, when you tell them, oh, wild type polio is eradicated, but you better keep vaccinating, they're going to say, well, we've got better things to spend our money on. So it becomes a challenge of how you maintain vaccination in a polio-free world for 20, 30 years till these guys have lived out their natural lives. And as soon as you stop using the oral polio vaccine, you should, in principle, stop creating any more of these people. But while you are using that, you risk making more of these, which then means you've got some time after eradication, while these guys are still in your population, that you can continue vaccinating the children. I would think it would be incredibly risky to stop vaccinating children in the post-eradication era. So unlike smallpox, where we could just stop, we're going to have to vaccinate against polio for many, many years to come.
Go to the back, maybe, and come forward.
Hello, I'm Sushant. I'm from India. Thank you for the wonderful presentation. I think besides the content, the way you presented was also very interesting. So I'm a public policy student here, so my question is more towards the policy aspect of it. So you mentioned that for Ebola virus, the pace and the investment which has gone in trying to develop a vaccine for Ebola virus has kind of doubled over the last few years. So my question is, like, we have a virus like Ebola, and then we have, let's say, diarrhea. So diarrhea claims, I don't know, like in India itself, it claims almost 200 to 300,000 lives a year. Yeah. And Ebola, which claimed few thousand lives in last year, and then 20 years back, 30 years back. So as comparing these two kind of viruses, so I think rotavirus vaccine has been in development for almost 20 years now, and before it reaches implementation in India, it's I think at least another five more years before it actually gets included in full immunization program. So what goes on in the decision-making process of why unanimously we all agree that we should double the pace of developing a vaccine for Ebola, whereas diarrhea is still lying in abeyance for almost 20 years now?
Diarrhea is a neglected disease.
Diarrhea is neglected, you're right. There are various factors. Some people argue that for diarrhea, forget vaccination, ensure clean water, right? Deal with clean water, provision of clean water to communities, you'll deal with the diarrhea problem. It's not the case for things like rota, where we still, in Western countries, get a lot of rotavirus diarrhea, but for some of the bacterial infections, like Shigella dysenteriae, for example, and cholera, and some salmonellas, the clean water helps enormously. The other thing in the comparison with Ebola was dramatic in its appearance. These hemorrhagic diseases are pretty horrid. No one dare go near them. People were left to die in isolation because of the fear of being infected. The death rate was 40%, 50%, and quite frightening for those that were infected. And it had the potential, it looked as though it was rapidly spreading throughout Western Africa and could have gone further. So it did cause an emergency response. You're right to say that diarrhea is perhaps even more important in the number of deaths, and we don't respond in the same way, but it's been there all the time. And therefore it's not a new emergence that galvanizes the world. The other thing to say about diarrheal disease, it's complicated. To develop one new vaccine is, like I said, expensive and takes time. And Ebola apart, I mentioned $1 billion in 20 years. For diarrhea, you've got Shigella, of which there are at least a dozen different serotypes. You've got Salmonella, you've got Campylobacter, you've got enterotoxigenic E. coli. So there are a wide range of organisms that cause diarrhea, and you sort of need vaccines against all of them, all to be individually developed and then somehow pooled together to vaccinate against diarrhea. So it's not, it's a bit like saying, can we vaccinate against respiratory disease? When you know you've got flu A, flu B, coronaviruses, rhinovirus, respiratory syncytial, paramyxoviruses, etc. It's a whole range of things that cause diarrhea, so it's not easy to tackle the whole thing of diarrhea. Yes, you can do bits like rota, where you tackle a piece of it, but mopping up and getting all of it is a hell of a challenge for a vaccine development program.
Questions on that? Can I just follow up on that? Do you think it's also a question that diarrheal disease doesn't have a lobby because there's so many different pathogens which cause it? There's no scientific lobby and vaccine lobby which is saying we must eradicate this condition.
Because kids don't vote.
Sorry?
Because kids don't tend to vote.
Yeah, I'm not sure that it's as cynical as kids don't vote, because if adults who lose a child, they vote. So I think there is the political will, and UNICEF and Gavi and WHO are fully aware of the burden of of diarrheal diseases. And clean water, as I mentioned, becomes an important provision for developing countries to avoid it. And the vaccination thing is just complicated. That's really what I was trying to say. We have another question at the back.
Hello. I was interested in your comments on the build-up of antibiotic resistance. In this country, we seem to have got it a bit. The Chief Medical Officer, Dame Sally Davis, spoke recently.
Yes.
Close to here about it, well attended. And NICE has just released some documentation on stewardship, antibiotic resistance stewardship. What point really is any of that when we still have countries not that far away from here where antibiotics can be bought over the counter and they're still being pumped into farm animals and the like? Is there any point us doing what we're doing when other countries are so lackadaisical with their stewardship?
Well, I think there is a need for a greater worldwide responsibility for the use of antibiotics, both in treating human health and, of course, in the farming industry. The push for new antibiotics is about having things that are not available to the general public or not available to the farming community and wouldn't be available across the counter, certainly, or at least not legally so. So you have let's say a fallback if you hit resistance. It isn't easy. I mean, one of the reasons for the dry up in the provision of the pipeline of new antibiotics is the economic model. To develop a new drug costs rather like a vaccine, but probably more, $1.5 billion, $2 billion. The first thing that the doctor does when he's faced with a bacterial infection is reach for penicillin or amoxicillin or tetracycline, which are pennies. He's not going to reach for the new antibiotic, which will have to be priced at thousands to get the billions back of investment. And in any event, the more he doesn't reach for it, the less will be used and the higher the price will have to be. So getting your money back as an investor, and unfortunately we live in a capitalist world where this rules what people work on, it really demands very, very high prices and widespread use of an antibiotic in order to make that investment worthwhile. And if you're not going to get that, which seems unlikely, you're not going to develop the new antibiotic. And that's been part of the problem. Now, there are mechanisms which we can use to overcome that, so-called pull and push mechanism, guaranteed purchase, guaranteed pricing for when the antibiotic arrives, push by pushing funding from government funds into the research and development. That means we can have a go at getting new antibiotics. The example that I gave was just one of a number that I found particularly interesting because it was based on this idea that there's a lot of places we haven't looked yet for natural products which could be antibiotics. But you're right, we've got to be more prudent in the future. We've got to be careful about how we use these antibiotics, or we've got to be very good at bringing new ones on rapidly. If living through that sort of 50s, 60s and 70s era, we didn't worry too much because we just reached for the next one if we hit resistance. Now that's getting a bit more difficult with these. As I mentioned, methicillin-resistant Staphylococcus aureus and extensively resistant tuberculosis. Those are problems.
Question there. A two-part question. Some people believe that a major pandemic is inevitable. I wondered what your opinion was on that. And secondly, considering the problems of bird flu and swine flu, etc., how big a problem are the intensive systems used within factory farming across the planet?
Well, inevitability is a sort of, it's a bit like infinity, really. Depends how long we're around. But if the human species survives long enough, almost certainly there'll be a new influenza pandemic. Through the last century we saw them, well, we had 1918, 1957, 1968, 1976, and then more recently in 2009. So, 4 or 5 per century. The most important was 1918, with an estimated 40 to 60 million people died. But Asian flu of 1957 also caused a lot of deaths, probably at least 4 million. And Hong Kong flu as well caused a lot. So yeah, we will see another pandemic almost certainly. The timing of that is incredibly difficult to predict. The severity is also difficult to predict. The last one, the Mexican H1N1, turned out to be a bit of a damp squib, fortunately. But another one like the 1918 could be dramatically devastating in certain countries. Would impact trade, would impact behavior enormously. I saw an estimate from a economist analysis suggesting that a 1918 type influenza pandemic would have a much bigger impact on the global economy than the 2008 financial crash did. So these things are not trivial.
They're worse than a banker.
They're worse than bankers, these damn viruses. So I think we will see it almost certainly. The question now is, can we get prepared? And how do we prepare? Stockpiling Tamiflu was sort of raised as a bit of a waste of money, but if I were in government, I don't think I'd dare not stockpile Tamiflu if I thought a pandemic was on its way, or even potentially likely. Vaccine manufacturing capacity has gone up enormously in the last decade. It still takes time. It still takes, we've got to prepare the strains, distribute them, and so on. In the meantime, the epidemic may be ravaging through the population, but our capacities are much, much bigger than they were probably in billions now, rather than tens and hundreds of millions as they were in the past. But the population is getting bigger as well. Now, as for the farming methods, interesting point that I'm sure impacts, but how big is that impact? Not sure. The 1918 and the probably the 1957 came without that impact. We haven't seen enormously more during the sort of intensive farming period of agriculture, but I'm sure it doesn't help.
Question here?
Shall I show?
Yeah.
No, because online they won't be able to hear you.
How do we tackle the anti-vaccine lobby? Because those people who die or are injured from measles today, that's completely could be completely alleviated. It's something sociological rather than anything scientific.
Yeah.
And can we apply that to the 3 countries today that have polio?
Well, first of all, the polio is probably not due to anti-vaccine in quite the same way. We have, of course, seen politically, yes, we have, of course, seen killing of the nurses that have been administering the vaccine. There has been some Islamic resistance to vaccination. That has meant a failure to eradicate it from those countries, or potential re-emergence in those countries. So it is a problem, but we've still managed dramatic declines in those countries from what they were in 1988 and earlier. We've got to keep the pressure up. The general question on anti-vaccines is about education, really. It's about being able to weigh the evidence, be convinced, evidence-based judgment, and really by getting the message out there. It is also reasonable to acknowledge that vaccines, because they're biological entities, and some of them are live, they provoke immune responses, they provoke inflammation at the sites of injection and administration. They're not totally without side effects. You might get a swollen arm, you might get a fever, you might get a chill, and there are also occasional rare things like Guillain-Barré syndrome, which is a complement problem that can lead to temporary paralysis and other clinical features. So it's hard to say that vaccines are absolutely 100% safe every single time for every member of the population. The point being, though, they are much, much lower in terms of incidence of any unwanted effect than the infection itself. And so the risk-benefit is hugely in favor of using the vaccine. But getting those sorts of messages across to the general population is sometimes difficult. There is also a little sneaky bit in here, which is if you all you lot get vaccinated, the virus doesn't circulate, therefore I'll be okay, even if I'm not vaccinated myself. So there's a bit of selfish behavior in this as well that we need to overcome. I'm not a sociologist, but I welcome whatever efforts are made in this area to try and minimize the impact of that anti-vaccine lobby.
I've got one question, which is, sorry, somebody else?
There's one at the back.
Oh, I'm sorry, one at the back. I won't ask a question then. Step to one side.
This is slightly more political rather than necessarily related to infectious disease, but in the news at the moment, we've got the recent mass migration from war-torn areas in the Middle East, and obviously people coming from Pakistan and Afghanistan, where you've got reservoirs of polio and other infectious diseases. And you've got mass migration through and into Europe. What do you see as implications for public health given the lack of sanitation and control of mobility?
Well, as I mentioned when I talked about Shigella dysenteriae, wherever you get refugees, wherever you get people in poor sanitation environments, you risk particularly outbreaks of diarrheal diseases, but also respiratory diseases as well. For polio, fortunately, any movement of polio out of that part of the world that might be linked to Afghanistan and Pakistan will be people arriving into a highly vaccinated community. So unlikely that polio will be a problem coming with migration and that poliovirus will spread. All our kids get, multiple shots of either killed or live vaccines, in some countries both. So that one I think doesn't concern me greatly. Diarrheal diseases will be a problem in the group of migrants themselves while they're together in crowded environments, but ought not to spread out because of normal hygiene and the way we can administer that. And then beyond that, things like hepatitis C, hepatitis B, HIV are not significantly greater, as far as I'm aware, in those communities than we have anyway. So at this point, I'm not seeing any particular infectious disease risks that are coming with those migrants. If we can keep those migrants themselves free of disease by making sure they get rapid access to healthcare measures, if there are any outbreaks among them, I think it seems to me we can probably do quite well in keeping the lid on anything that may arise.
Jeff, I think that's a good time to end and a good time to say thank you again for a wonderful presentation. Thank all the members of the audience for their questions, and we can retire for drinks now. So thank you.
Thank you.